Good evening, everybody. So I just wanted to give you some highlights of the material that we're going to be covering this week. When you're going through the 202 material, you'll notice that the lectures that we have recorded are very similar to what you have in the textbook. The new textbook is the Mary of Home textbook, and obviously the content in there is going to be similar to any other textbook that you would have for A&P, including the old one that we had, which was Tortora. So the lecture material that you have online is actually, in terms of the figures, etc., based on the Tortora book, but there will always be analogous figures and tables in your Mary of book. Now for these highlights, I'll do these on a weekly basis and just kind of hitting the high points, making sure that you spend extra time on particularly hard things or whatever, and I will use the Mary of textbook figures, etc. for those, if that helps. Alright, so again, these newer sessions are not to substitute for the lecture material, but in fact are just, like we said, kind of the highlights, the main important points that you want to see. So I'm going to flip through fairly quickly, knowing that you're going to have a full lecture or more on this material, but as I flip through the slides, I'll recommend particular things that are important for the exam or particular things that are important for you to focus on and learn. Okay, so as we get into blood, this table right here is definitely a good one that goes through and highlights the different components that we have of the blood. So we have the cellular elements as well as the fluid components. Sorry, I'm just kind of organizing some of my writing utensils, etc. So over on the left hand side here we have those things that we find in the plasma. Over on the right hand side here we have what we call cellular elements. And so it is important, the content that you see in here is very important. You need to be able to identify different molecules and ions, etc. that we find in the plasma and what are they there for, as well as the different types of cells that we can find in the blood. And when we say cells, this includes the cellular elements that are not entire cells, like the platelets. And then when we get to the leukocytes, the white blood cells, you do need to know the 5 different types and specific functions for each. Okay, so general characteristics about blood and what it does. So that's certainly important information as well in terms of what it's transporting around, etc. The details of a lot of the chapter goes through these particular cell types, their anatomy and their physiology. You'll talk about the different parts of the red blood cells, the hemoglobin molecule in particular, and how it has these different heme groups, which at the core have iron, which will bind up to 4 oxygen molecules. And here you can see that iron, right? Fe is a symbol for iron. So each hemoglobin molecule that's contained within the red blood cell, within the membrane of the red blood cell, can hold 4 oxygen molecules. Okay, so we talk about its role in terms of holding on to and delivering oxygen, as well as other things as well. It can bind carbon dioxide, hydrogen ions, nitric oxide, and things like that, even carbon monoxide. For any of our different cell types, we do need to know how they're produced, what their precursor cells are, and then at the end of their pathway, how do we get rid of them? Because they're only going to survive a certain amount of time. So you should also know their duration. So for instance, red blood cells are only going to be alive and circulating for about 120 days compared to some of your white blood cells that could be around your entire life. Platelets as little fragments will actually be around very, very short periods of time, on the order of days or maybe weeks. Okay, so again, we do talk about the development of the different cell types, the stem cells or the precursor cells, and then some particular stages. And there's a table coming up that actually goes through and shows you all the different cell types and the stages you would need to know, like for the red blood cells, the immature blood cell that's called a reticulocyte. Okay. In terms of diseases and disorders, you should know the different ones listed in the exam objectives. So for instance, polycythemia and anemia. You should know hypoxia and the different types of hypoxia. Talk a little bit about blood doping. And as we mentioned, knowing this whole pathway of how red blood cells or any of the blood cells are maintained. What's the signal to the body to maintain or create more of them? How do we get rid of them, etc.? Usually we're talking about hormones that are going to be involved in stimulating their production. Then at the end we end up recycling some of the components of the cells. It's kind of a green way that our red blood cells are used. And then we get into the white blood cells. Again, 5 different types. They separate into 2 different groups, the granulocytes and the agranulocytes, each with a different name. We'll look at them in lab as well. You need to be able to physically differentiate between them, but then know their different functions also. Never let monkeys eat bananas will give us the first letter of the names of the 5 different types of white blood cells in order from most numerous to least numerous. So we'll talk about that in lab. And then we basically get into the different cell types. So for instance, the neutrophils, which are like the bacteria slayers, the eosinophils, the basophils, and then within the agranulocytes, we have the lymphocytes and the monocytes. Alright, so all of these different white blood cells are associated with defense, so you do go through a bit about how they defend us. We'll talk about that more as we get into the immune system. So we're just introducing these white blood cells and their role in defense in this chapter, in this system, but we'll get into the details of it, more so in the immune system later on. So again, this is a good table that goes through the different formed elements or cellular elements, and key components are certainly their function as well as their anatomy, their names like you see in the first column, and what they look like. And here's one of those tables, as we mentioned, that give us our stem cell lines. We go from a hemocytoblast, the precursor cell like we see up here, which is also, when they talk about it as a megakaryocyte, it differentiates into 2 different types of stem cells, myeloid or lymphoid, and then ultimately you want to know what those different cells differentiate into. Lymphoid stem cells give rise to lymphocytes, and then myeloid stem cells give rise to all the others. Platelets, as we talk about platelets, that's going to get us into the idea of what's called hemostasis and how we keep the blood inside of us. There's our megakaryocyte again, like I was mentioning. And this is a good table that shows in all the squared areas that you see there, the different stages that you should know. The terminal stages at the bottom, but also these other precursors. Any of the ones that aren't in a box you don't have to worry about. Okay, so in terms of hemostasis, you should know the 3 phases. Vascular spasm, platelet plug formation, and then what's called coagulation or blood clotting. Blood clotting itself is a bit more complex than just saying the phrase. There are a number of different factors that are involved that lead to actual blood clotting, and you see that over on the right hand side here. And in fact, there are 2 different pathways. One's an extrinsic pathway, which occurs outside of blood vessels, and the other is an intrinsic pathway, which occurs inside of blood vessels and is a full pathway of different proteins and enzymes, ultimately, in either case, arriving in what you see down here, activating fibrinogen, which is a plasma protein, and causing it to become insoluble and form these fibrin threads, which solidify the clot. Okay, so you should be able to compare the intrinsic and the extrinsic pathways for coagulation or blood clotting. Other things you should know: if we know how to form a clot, you should know how to get rid of a clot, which involves this plasma protein, which is called plasmin. Very interesting story about how that works. And then towards the end we talk about how things can go wrong as well. So the formation of a blood clot, an erroneous blood clot or a thrombus, if it starts moving, how it becomes an embolus, and how that can be prevented using a variety of different medicines, as well as your body's own different responses to try to keep those things from happening. Other important things that we see out of this chapter are the blood groups. You should be able to describe the differences in terms of antigens and antibodies between our 4 different blood types. So we can see them either in this figure here or in this figure here taken from the book. So 4 different blood types, AB, B, A, and O. What does that mean in terms of the antibodies that are found there in that blood type, as well as the antigens or the agglutininogens that are found attached to the cells? Rh factor is something you also should know, and something that we'll actually discover as we get into lab. And this is just another table like the one you saw before that does a good summary of our different cellular elements as well as their particular functions. Okay, so in terms of the blood, this is not too bad. We're talking about the tissue that's inside the cardiovascular system. So in terms of complexity, a tissue isn't that complicated, but we did hit the highlights of things that you need to know for this fluid and the cells that are found inside of it. So next we'll be working our way into heart, the main organ of the cardiovascular system, and then into blood vessels as we continue on. But this will do it for the highlights for the blood chapter.